Hello and welcome to Naked Genetics, the show in which I try to find out just what's going on in your genes. I'm Will Tingle, and join me as we seek to cover the latest in genetics research and see just what the front line of genetic mapping and sequencing can reveal about our world. In this episode, how much of your personality is down to your DNA? We also look at synthetic DNA and why you'd want to make it, plus the spiders that turn one species of insect against its own kind. First up this week, let us delve into a couple of new pieces of genetics research and get our in-house genetics experts to give us their say on it. It's time to link back up with my gene geniuses, Shivani Shukla and Aylwin Scally. Let's kick off with the first one, which asks the question of how much of your personality is decided by the composition of your genes. A new study in the journal Nature Human Behavior has shone a light on the relationships between the genes that code for what scientists refer to as the Big Five personality traits. In doing so, they have looked at their interrelationship of personality traits and their relationship with other complex human traits to see how these genes affect how we think and behave. Shivani, to kick us off then, what are the Big Five personality traits?
So the Big Five is based off of the FFM, which is an accepted psychological framework. So under that comes extroversion, which is risk-taking, openness, agreeability, neuroticism, and conscientiousness.
And prior to this, had any of them been more studied than others?
Neuroticism, which is basically how likely someone is to engage in negative behavior traits such as anxiety and depression and those sort of things, that was very heavily focused on. So most of the previous literature has linked genetic loci with that trait, but this was the first study and is actually the biggest study of personality traits and have focused on the other four a lot more.
It seems like an absolutely vast endeavour and undertaking to try and map so much of the human psyche. What did this study do to try and do that?
It was a huge data set. There were 70,000 individuals, and it's another example of a GWAS study. So you take all the genetic information from these people and you try and map loci, which are variations in the genes. So all these 70,000 people had answered personality quiz questions and had self-reported the degree to which they're each of the five personality traits. And then once their DNA was sequenced, they mapped the traits to certain loci. And then that's how they kind of associated X with Y and found, what loci are associated with each of the five.
How did it find that these five traits interacted with one another?
They kind of found for each trait, what does it increase the person's likelihood of doing certain things. So, for example, people who had loci for openness were more likely to be artistic and pursue educational opportunities. People who were conscientious were more likely to engage in day-to-day physical activities such as DIY, and being risk-taking is linked to displaying neurotic behavior, but being agreeable actually is a negative predictor for displaying neurotic behavior. So it was quite an interesting mix of what you're more likely to do in your day-to-day life and then how each trait links to the other.
Aylwin, to bring you in, given how much we love the discussion of nature versus nurture, does this provide any new revelations as to how much of your personality is hardwired into your DNA?
On the face of it, that seems like a very unsurprising result to say that people who are more agreeable are less likely to be neurotic. What one certainly can't say, I think it's really important to reiterate again with all of these studies, how much of the variation in these traits is actually being accounted for by genetics or is correlated with the genetics. It turns out it's about 5% in this particular study.
Yeah, that figure of 5% really does hammer home just how much of people's personalities has to be down to their life experiences and their nurture. I mean, the genes encode for a rough map of brain wiring, and then everything you are and everything you go through kind of molds the map into the final specimen. I think that's fair to say. And it would be extraordinary for me to be able to look at someone's genes and go, they've got one gene here and they've got one loci here. They must be an agreeable person. That just doesn't sound possible to me.
Indeed, and that's definitely not what we've discovered here.
What stands out to me is that given that this is a study that is almost entirely self-reported, it seems a very subjective way of trying to quantify how different individuals think and act.
Without doubt. And there's obviously the reporter bias, there's the bias of veterans perhaps not being representative of the general population. This data set was taken from veterans, so personality is definitely subject to change over a lifetime. And since it was self-reported, veterans are perhaps not accurate representation of the entire population.
Yeah, I mean, we should also add, you end up sounding pretty negative if you say, well, really, you're showing a very small amount of variation in this trait explained by genetics or accounted for by genetics. But of course, the interesting thing in many cases, in many such studies, is which bits of the genome are implicated. Are there proteins or genes that we can potentially target for drugs in psychiatric care or in other circumstances? Does this help us when we're trying to understand complicated social or psychological interactions or disorders? In other ways, we can use these genetic correlates as a sort of a quantitative handle on those traits in other research. And I think that's the real purpose of doing these things. It's not really to show, yes, genes can affect some traits in your body or in your mind.
Yes, I think, as you well put out there, it would be far more interesting for me to be able to look at someone's sort of genetic makeup and go, they have, however small it is, a predisposition towards neuroticism due to the presence of this gene. Can we help them treat this?
Yeah, and actually from the study, they found a certain gene which is linked with the hypothalamus pituitary adrenal gland pathway. And by kind of linking, it's a receptor of a hormone. So by linking that to neurotic behavior, it's kind of been identified as a good target for psychiatric treatment. So I think it's absolutely right. The application in terms of what we can do with it from a medical or treatment sense is probably more important than like a very expensive and detailed personality quiz.
Thank you very much, Shivani. We look forward to seeing what you bring to the table next month. But now it is over to you, Eowyn. Rapa Nui, also known as Easter Island, is one of the most isolated islands in the world, 3,700 kilometers away from the nearest continental landmass of South America. The story of the inhabitants of Rapa Nui has long been presented as a cautionary tale of environmental exploitation, as it was believed that their rampant use of the local fauna and flora led to a resource shortage, followed by conflict and cannibalism, and subsequent genetic collapse sometime around the year 1600. But a new study, just out in the journal Nature, disputes that theory, saying there was genetic evidence pointing towards a healthy population throughout their history. So I guess the first question is, why was there ever an ecocide theory in the first place?
In this case, I certainly think, even as a child, hearing stories about Easter Island, as it was called then, and how there had been this terrible collapse due to them using up all the resources of their island and leading to wars and even cannibalism and a massive population collapse. I think most probably it was the subject of a book by Jared Diamond about 20 years ago, where he probably did a lot to repopularize or to disseminate this idea, this ecocide story. It's important to say that we're at risk of propagating another sort of mini myth here, which is that genetics is coming along and constantly disproving the archaeologists and confounding their myths. Actually, there's been lots of evidence presented before this study involving archaeological evidence, not genetic evidence, which has cast a lot of doubt on this theory. So I would say that actually already there were many reasons to think that it wasn't true, but genetic evidence has provided another very important piece of evidence against it.
How was this helped to be further disproved with genetic studies?
So there's two things really that you can look at with a set of genetic evidence, and they looked at some what they're calling ancient Rapa Nui individuals, but actually this data probably came from individuals who may have lived as late as the early 19th century or mid-19th century. Some of them, in fact, are from the 20th century. And what they did essentially was they looked at two things. One was, could they see any evidence of the impact of a population crash on these in the time frame that had been proposed? Could they see any evidence of that in their ancient samples? And they also looked at another big question about Rapa Nui, and about Polynesia in general, which was contacts with South America. That's another interesting story. Taking the second one first, they were able to see signals of ancestry from that was similar to the ancestry that you find in Native American populations in South America, indeed, not just in South America, but in fact on the west coast of South America. They find ancestry that is similar to that in these Rapa Nui individuals. And you can actually try and date by looking at the distribution of those ancestry segments, you can actually try and date when did they appear in that population. And that contact maybe goes back about 1,000 years. So long before the arrival of Europeans in Polynesia, the ancestors of these individuals had actually managed to get all the way across the Pacific Ocean and indeed have contacts such that people were coming back from South America to Rapa Nui and other islands. So that's really fascinating. The other side of the story about the ecocide, well, that's a little bit harder to analyze, because there are various types of things that you might call a population crash that might have that may or may not have a strong signal in the genetic evidence, and they looked for evidence of a certain particularly strong population crash along the lines that people had argued that the population fell to only 10% of its previous value within a couple of generations. They looked for evidence of that, and they couldn't find any of their models that fitted that data very well. What their data fitted much better was just a steady, slow increase in population since from a sort of a founding population maybe about 1,000 years ago. Right up to the time of the 18th century, just a slow growth, no particular evidence for any sort of massive crash. That's not to say that there weren't lots of other things that did happen. There clearly was deforestation. I mean, we can see that, and there may well have been wars, and there may well have been famine and all kinds of other things, but none of those things necessarily lead to the kind of emptying out of the island that this ecocide story seems to presuppose.
So not only has this disproven the previous ecocide theory, it almost goes the other way, because there's far more unexpected visitors in their genetic lineage than perhaps was anticipated.
Yeah, I would argue that there's no particular evidence to assume that this was a population in decline. There may well have been lots of terrible things happening. And so there are some aspects of that story which we should be careful about what we're saying is getting disproved here. Just in narrowly sort of demographic terms, there was not some sort of terrible collapse happening. But again, just to come back to this point, you could have things which would be perceived as quite severe demographic events in the sort of oral history or in our historical memory or historical record, such as 30% of a population being wiped out within a generation, something like that, which would actually be quite a dramatic event in terms of our understanding. That actually wouldn't necessarily have a big impact on the genetics of a population. So there are things which there might well have been events like that which could have happened. I'm not saying that there's any evidence at all for those things actually in this particular case, but there are other cases around the world where it's not so straightforward to match up genetic evidence with historical records or accounts that might exist about such events.
Eowyn Scally and Shivani Shukla, thank you very much indeed. You're listening to Naked Genetics, the show about genes and the geniuses that study them. Now, for the past few months, I've been linking up with genetic sequencing company Illumina to find out what DNA sequencing can reveal about our world. And up until this point, we've been taking somewhat of a passive approach to our DNA sampling, very much taking the DNA as it comes. But what if, just this once, we took the initiative? What if we made our own DNA? Because you can just do that. You can chuck a load of C's and G's and what have you into a bucket and give it a stir. So what would you end up if you did? Well, here to explain synthetic DNA is Camina Biosciences' James Richardson.
So synthetic DNA is DNA which has been derived from artificial means. So it's not something that's being part of an organism necessarily. It can be, but ultimately it's something that is not a directly natural product, whether it's chemically made in a lab or through other means to make artificial sequences of DNA.
So, the big question, how does synthesizing DNA work?
Historically, there's been one way of really making synthetic DNA for quite some time now. So for decades, it's been a process called phosphoramidite chemistry. This is a purely chemical process, and so it involves various chemical steps through which you add one base at a time.
And look, I can't sugarcoat it, creating DNA is actually a complex process, and we could get lost in the weeds with this chemistry, believe me, I did. But let's leave it as phosphoramidites are used to construct a tailor-made chain of DNA bases, linking one after the other to create a ladder-type structure, such that you can put it in pretty much any order you want.
Fundamentally, there's not actually a difference between synthetic DNA and natural DNA once you've made it. The 4 bases are identical, and really that's the aim of almost all synthetic DNA, to mimic the natural DNA, except we just have control over what sequence is being made.
Okay, bit more than a few base pairs in a bucket, as it turns out, but given the complexity of the procedure, it almost seems flippant to ask, what's the point of synthetic DNA?
Synthetic DNA has a number of applications, some of which are like protein engineering, so that could be for things like therapeutic applications. There are a number of applications in the sort of agriculture space as well, and also now with gene therapies. And so there was a recent trial of a gene therapy in which deaf children were treated with this new gene therapy and have now been able to actually recover some of their hearing. Essentially, you're replacing a faulty piece of DNA that someone may have with one that's got a correct sequence. And so if you can then do that in a manner of a targeted way, so that you can replace these broken parts of someone's genome, you can then begin to reverse the effects of some of these diseases.
Truly extraordinary that we are approaching an age of being able to sub in our own DNA to replace faulty strands. But let's keep our eyes on the prize here. We're here to sequence DNA, even if it is synthetic. So let's meet today's lucky sample, introduced by Illumina's Ilaria Grisi.
James has kindly given us a sample of synthetic DNA, and we have also ordered the equivalent natural DNA, which has been extracted from a phage.
Yeah, so phage is an infectious organism that will infect bacteria, and it's commonly used within research as a mechanism for sort of delivering DNA into cells.
A short line of DNA, about 1,800 base pairs long, has been selected from this phage's back catalogue. This is our expected DNA, the series of bases you'd expect to see in this strand. A synthetic version has been made, following this recipe to the letter. But to check how it stacks up against Mother Nature, a sample has been taken from a live phage to compare and contrast how the two stack up against each other. It's man versus nature at a microscopic level.
And they were almost the same, but not quite, actually. We found 3 differences between the 2 DNAs, and it turns out that the synthetic DNA that James' team has done was actually the perfect copy of what it was intended, and it was the natural DNA from a phage that actually had 3 errors in it. So I think the chemistry is perfect in copying nature, but nature is not always perfect in copying itself. So I guess this is how evolution came about.
So with 3 errors out of 1,800 base pairs in the natural DNA compared to the synthetic's 0 errors, they've actually made a more accurate and more stable copy of the phage's DNA than the phage itself. Sequencing it in this manner is a strong form of quality control, but it also may have other uses.
As James was mentioning earlier, we talked about a specific vaccine for your specific cancer. There is also, as you remember, during COVID, we had all these different variants. All this COVID variant were effectively different in DNA sequencing for the different strand of the virus. So it helped us understanding how the virus was evolving and making sure that the vaccine we had in place were always well suited to the particular variant.
The idea of personalized medicine and making sure that we're applying the right therapy for the individual is a major area for both sequencing and synthetic DNA.
But let's not be too harsh on nature, given that each organism forms and checks its own DNA instantaneously millions and millions of times a day. It's a testament, really, to just how remarkable evolution is in that regard.
And this is why nature sometimes makes mistakes.
A great thing, then, that our work is sometimes riddled with errors, or else we'd still be single-celled pond dwellers. I tried that line with the higher-ups here, but it didn't go down well. Still, though, a very exciting time in the world, and one last thing to think about. Given that we can now make our own DNA very well, and given just how much information is stored in DNA, could we one day harness the power of DNA as an information storage device?
If you take the examples in nature, it's there to some degree in that we can extract DNA out of preserved samples from thousands of years ago and actually get good sequencing results out of it. And so from an information storage, it's already happening in nature. I think there's a fair amount of work going into looking at using it from a synthetic side now as well. It's definitely a hot area of research.
I mean, it's really exciting to think about how efficient a storage system that is, how much information is coded in so very little space. and that can be made and preserved without using too much energy. So it's certainly a very, very exciting prospect, but I do believe that all the research are very, very early stage, so we are not really at the point where we can even have a prototype of a storage device, I believe.
Some way off a DNA USB stick then.
Yeah. No.
Ilaria Grisi and James Richardson, thank you very much. And so we come to the end of our time with Illumina. Thank you to everyone there and elsewhere that took part in Will It Sequence. And who knows, maybe one day we will delve back into the world of sequencing once more. But for now, to round off this month's Naked Genetics, it's time to explore another quirk of evolution. You join me at dusk in the Hubei province in central eastern China. It's a part of China known best for the Three Gorges Dam, the city of Wuhan, and thousands of lakes. In summer, temperatures sit around 24 to 30 degrees Celsius with a decent dash of humidity thrown in too. Very pleasant conditions for one of my favourite organisms, the firefly. And who doesn't love a firefly? You can look at them, you can put them in a jar and read a book if you wanted to. That's pretty much it. But there is one species in these here hills that look at these ethereal, enchanting lights and sees the opportunity for an easy meal. In steps the orb weaver spider, Araneus ventricosus. It's an unassuming creature, grey-brown body, a few centimetres in length, scattered across much of Eastern Asia. Visually a bog-standard spider. Every evening it goes about the business of forming its web and hunkering down for a night of sky fishing. But the most successful fishers use a lure. Fireflies aren't just flashing because they can. I certainly would, but that's probably why I haven't been blessed with such power. No, firefly flashes are a means of communication, and obviously that includes communication between males and females. It's also apparently used to warn away bats in some species, which is pretty wild, but a story for another time. Fortunately for us, it's a very simple system. If you are a female, you flash once. If you are a male, you flash twice. And naturally, females are often inundated by many would-be suitors once they reveal themselves. And I'm sure most of you are ahead of me by now. A firefly gets tangled in the spider's web, but instead of consuming it straight away, the spider waits, because the firefly continues to flash. But this is where it gets odd. Researchers at Huazhong Agricultural University took a census of the fireflies found flashing in spider webs, and they're nearly all male. Given what we know, that doesn't add up. Why would male fireflies dash to the location of a fellow competitor? Well, for some reason, which cannot yet be fully explained, when stuck in a web and in the presence of a spider, a trapped male firefly changes its signal from 2 flashes to 1. Is this one last act of spite from the doomed male? If I can't live to mate, no one can? Is this a very unfortunate case of mixed signals? Perhaps the firefly for help is a very similar flash to that of I'm a female, let's mate. Probably no to either of those. In fact, the most prominent theory is that the spider is manipulating the fly. The researchers postulate that the spider venom actually changes the flashing pattern of the male, but just how that works is unclear, and what a cosmic miracle it would be for a random venom to be the perfect means of luring in more dinner. Behaviour alteration isn't unheard of in the animal kingdom. That's why rabies victims suddenly become more bitey, and why that stray cat was suddenly so friendly all of a sudden. But these are from internal parasites, not from external sources. And also, is this technically tool use? Using an external object as a means of manipulating the world to the wielder's benefit? That's a remarkable leap in cognitive function for a spider the size of a golf ball. For now, this act of master manipulation remains a mystery. But next time you're lucky enough to see fireflies, just remember, they aren't always what they seem. That's it for this month's edition of Naked Genetics. Do join me again next time for another series of Genetscapades. In the meantime, if you are enjoying the show and you do wish to support us here at The Naked Scientist, do consider donating. We've made it very quick, easy and secure on our website, nakedscientist.com/donate. And if you do want to get in touch, my email is, of course, willt@nakedscientist.com. But for now, until next time, stay safe, thank you all very much for listening, and goodbye.